Good afternoon.
Good afternoon, ma'am.
Can you?
Yes, ma'am.
Okay. Again, only two students. Alina and Havar.
Yes, ma'am.
Do you know about other students?
We don't know, ma'am. I think so. They are sleeping or busy in their schedule.
Ma'am, most of them have left.
Okay, then we can start.
Yes, ma'am.
Ma'am.
Talk about which topic we will talk today.
Ma'am, structure of bacteria, function, detection of structure,
Uh-huh.
capsule,
Yes.
polyntine granules, and flagella.
Yes. Okay, who wants to start?
Yes, ma'am. Structure.
Yes.
Okay, ma'am.
Okay, Alina. You can.
Ma'am, basically, bacteria are prokaryotes lacking well-defined nuclei. They don't have nuclei and membrane-bound granules, or and with chromosome composed of single DNA. And they come many. Bacteria have many shapes, from shapes and sizes, from minute spheres, cylinders, spirals like thread, and also have rod shapes. There are usually four types, which we have already discussed in our previous lecture: coccus, bacilla, spiral, and vibrio. And they also form in the spiral coccus, rod like bacillus, curved in vibrio form. And it is a visual seen variations in shapes, and cells can be stretched in one dimension. And there are also other 10 types. These are 10 types most जो dangerous होते हैं, like E. coli bacteria and Mycobacterium tuberculosis. They are dangerous in some aspects. And we if we classified bacteria into five groups: same, spiral, rods, vibro, and spirochetes. And they can exist in single cell, in pairs, and in also chains, or in also clusters form. Basically, these are the structure of bacteria. Hello, ma'am.
Yes. Can you tell about uh, differences between eukaryotic and prokaryotic cell?
Eukaryotic and prokaryotic.
Yes.
Ma'am, basically, eukaryotic have defined nucleus with boundary, and prokaryotic also have nuclei, but it doesn't have a defined nucleus boundary. So we usually consider eukaryotes have nucleus, and prokaryotes have not nucleus. It's a main difference between a prokaryotic and a eukaryotic. Other difference also. Uh, uh, we usually have there are different types. Nucleus is where are stored in genetic information. So prokaryotes don't in also have prokaryotic or eukaryotic chromosomes. Prokaryotics contain single circular chromosomes, and eo has multiple linear chromosomes. And because prokaryotic DNA can interact with cytoplasm, transcription, and translation, because it don't have a boundary around it. And on the other hand side, eukaryote transcription occurs in the nucleus, and a translation occurs in the cytoplasm, because it has a well defined cell wall or cell membrane.
Mhm.
So some eukaryotic genomes are organized into operons, but most are not. And on the other hand, prokaryotes own only one copy of each gene. They are haploid in form, prokaryotic chromosomes.
Mhm.
And on the other hand, non-essential prokaryotic genes are commonly encoded in extra chromosome plasmid. And and in eukaryotic, extra chromosome plasmids are not commonly present in eukaryotic. And eukaryotic contains a large amount of non-coding and repetitive DNA, and prokaryotic are efficient, containing little repetitive DNA. And
Yes.
yes, ma'am.
Uh-huh.
On the other hand, we if we talk about their size, single cell wall, so their size single cells, and on the other hand side it's eukaryotic multicellular. Cell wall present comprise of peptidoglycan, and on the other hand eukaryotic usually cell wall absent. If present, plant cells and fungus comprise cellulose polysaccharides, or and mitochondria is present, and on the other hand side it's absent on the other. And shape, we already discussed about shape. It has a circular double standard shape. On the other hand side it has a linear shape.
Yes, you are
Yes.
right. The main differences in organella, yeah, which eukaryotes have mitochondria, Golgi
Yes, ma'am.
apparatus. In prokaryotes, they don't have a nucleus membrane. Okay, right. Так, Havar, are you here?
Yes, ma'am.
Can you tell about cell wall, structure of cell wall, and differences in positive and gram negative bacteria and function?
Ma'am, voice is not
Cell,
clear.
cell
Structure
wall,
of cell wall and
differences in
totally
cell
cell wall.
wall. Yes, yes.
Ma'am, a cell wall is a structured layer surrounding some types
Ma'am,
of cell.
you asked about him gram positive or gram negative.
Uh-huh.
You
Okay, ma'am.
asked him
First
gram
of all,
positive or
ma'am,
gram negative.
Yes, yes. Differences in cell wall.
ma'am,
Crystal
a
bacteria.
cell wall is a structural layer surrounding some types of cell just outside the cell membrane, and it protects the surroundings. And it can be tough, flexible, and something rigid. It provides the cell with the both structural support and protection, and also acts as a filtering mechanism. Yes, ma'am.
Yes,
And
continue.
gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane. And the
Uh-huh.
gram-negative lipid have a thin peptidoglycan layer and have an outer lipid membrane.
Uh-huh.
Yes, ma'am. Next,
Yes, another differences.
ma'am, and gram-positive bacteria have lots of peptidoglycan in their cell wall, which allows them to retain crystal violet dye so they can sustain purple blue. And on the other hand, gram negative bacteria have less peptidoglycan in their cell wall, so cannot retain violet dye, so they stain red pink.
Mm-hmm. That's why uh, 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 they can stain in different color.
Yes,
For
ma'am. And
example,
the gram negative bacterium Escherichia coli is the most numerous aerobic conventional inhabitant of the large intestine. For example, and the stain causes of diarrhea and all can cause infection when they invade style sites.
yes.
And the cell wall of gram-positive bacteria is composed of thick layers, peptidoglycan, and the cell wall of gram is composed of layer of peptidoglycan in the gram staining procedure. Ma'am, for the main difference is the gram-positive color retains the purple color, and gram-negative bacteria produce endotoxins.
Mm-hmm.
Yes, ma'am.
Yes. Okay, next
And
you can continue with cell membrane or
cell membrane.
Alina can.
Ma'am, I can, no problem.
Okay.
Okay, ma'am.
Ma'am, the cell membrane is also known as the plasma membrane or cytoplasmic membrane, and historically
Mm-hmm.
referred to as the plasma lemma is biological membrane that separates the interior of all the cells from the outside environment extracellular, which protects the cell from its internal environment. And the function, the plasma function of cell membrane, and it provides the protection for a cell. It provides fixed environments inside the cells, and that membrane has several different functions. One is to transport nutrients into the cells, and also to transport toxic substance out of the cells. And the
Yes,
cell
and
membrane cell membranes made include plasma membrane, and internal membranes are made of glycophospholipids molecules composed of glycerol, a phosphate group, and two fatty acid chains. Glycerol is three carbon molecules that function are backbone of these membrane lipids. And and
yes, yes, you are right. In
ma'am, here
yes,
it have long chains
Talk about cell
composition,
membrane of bacteria cell.
and other have prokaryotes. If we start lengthy, these are the qualities of cell membrane. It's ma'am, we can only tell about the cell membrane, its composition, its function, its structure only, no?
yes, yes,
And I had tell everything. The structure I
yes.
told structure, functions, and composition. Yes, ma'am. Next, if you want to ask something.
Next, after cell membrane, we have этот.
Ma'am, after a detection of structure, capsule, volatile granules, and flagella.
Yes,
Granules, they granules,
after cell membrane, let's talk about
lysosomes,
that. No, first cytoplasm.
cytoplasm, ma'am.
Yes, because see, first bacteria have
Ma'am,
cell membrane,
Yes.
that cell wall, then
yes,
cell
ma'am.
membrane, and
Yes, ma'am.
after
Their
this,
structures, organelles.
okay,
And
tell
then we
about
have capsule.
cytoplasm.
Ma'am, cytoplasm is a gelatinous liquid that inside the cell, and it composed of water, salts, and various organic molecules.
Yes.
And some intracellular organelles, such the nucleus and mitochondria, are enclosed by membrane that separates them from the cytoplasm. And the cytoplasm function in the cell. The cytoplasm is responsible for holding the components of the cell and protect them from damage. It stores the molecules required for cellular process and is also responsible giving the cell its shape. Basically, its main function is for giving the cell shapes, and it stores the molecules which are present in the cells and present and responsible for cellular process. And 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 I already told that cytoplasm is made up of water, and water is the largest component in the composition of water. Uh, and apart from water, it is made from sugars, inorganic salts, and other organic components. And the cytoplasm are also composed of various organelles, which also form the endomembrane system and cytoskeleton. And cytoplasm, ma'am, the organelles of cytoplasm are little organs that are suspended in the cytoplasm of the cell. Each type of organelle has a definite structure and a specific role in the function of cell. Examples of cytoplasmic organelles are mitochondria and ribosomes, endoplasmic reticulum, Golgi apparatus, and lysosomes.
Okay, Alina Shekhzadi, you can continue with organella.
Ma'am, which organelle?
Mesosome.
Okay.
Then after mesosome, there are
Yes, ma'am.
Granules.
Okay. Ma'am, mesosomes or chondrites are folded investigations in the plasma membrane of bacteria that are produced by chemical fixation techniques. used to prepare samples of electron microscopy. And mesosomes or this function, it helps in cell wall formation. It also helps in DNA replication and distribution to daughter cells. And
Mm-hmm.
they help in respiration, secretion, to increase the surface area of plasma, and in also in enzyme content. And it's a mesosomes usually are membranous bacterial organelle. And in prokaryotes, if we talk about the on the basis of prokaryotes, then in prokaryotes these are specific structures known as mesosomes. They are invaginations of the plasma membrane, and
Mm-hmm.
it's helping DNA replication, cell wall synthesis, cell division. And other on the other hand, mesosome bring information of this crosswalks or and septum attach bacterial DNA to the cell membrane. So it's why it's also called mesosomes, also called chondroids or folded in because it's folded in vegetations. So it's usually we used in fixation techniques. And it's, yes ma'am, I think so.
Mm-hmm. Okay, good.
And mesosome present, yes, main point, mesosome present in both gram positive and gram negative bacteria. So
Mm-hmm.
mesosome of gram negative cell typically lamellar and few numbers, on the other hand less extensively developed than those gram positive.
Mm-hmm.
Yes ma'am.
Yes.
And bacterial cells do not have membrane bound organelle like plastids and mitochondria. Their plasma membrane
Mm-hmm.
is however enfolded into mesosomes because it doesn't have membrane bound organelle, which enfolded into mesosomes, which carry respiratory enzymes and are known as mitochondria of bacteria.
Mm-hmm.
Yes ma'am.
Yes. Okay. Next
Yeah.
question. What is plasmids?
Plasmids,
Plasmids. Yes.
ma'am, it a small extra chromosomal DNA
Mm-hmm.
molecule and within a cell that is physically separate from chromosomal DNA and can replicate independently. So
Mm-hmm.
it is a double standard DNA molecules, and plasmid are sometimes present in archaea and also in eukaryotic.
Mm-hmm.
And if we talk about its a function, then it's a small circular DNA. So plasmid naturally exist in bacteria cell. So they also occur in some eukaryotes, and often genes carried in plasmids provide bacteria with genetic advantage such as antibiotic resistance. And if
Mm-hmm.
we talk about what exactly it is, then it is a small circular DNA, so found in bacteria and other cells. And
Mm-hmm.
plasmids are separate from bacterial chromosomes, and so that's why it's independently replicate, and only a small number of genes, notably some associated with antibiotic resistance. So it's if we talk about its replicate or not, every plasmid has its own origin of replication. So that's why we say that plasmids independently replicate, and so there can be many copies of plasmids, even hundreds within one bacteria cells.
Mm-hmm. Yes.
Yes ma'am. And if we talk it's beneficial for humans or not, plasmids are used by their host to cope with the stress related conditions. And many plasmids, for example, carry genes that code for production of enzymes to
Mm-hmm.
inactivate antibiotic or poisons. And other contains genes that help in host organism digest unusual substance and kill other types of bacteria. So we can say that it's beneficial for humans because it's causes a resistance to poisonous and cope with stress related conditions.
Mm-hmm. Okay,
Yes
good.
ma'am.
Yes, that is extra chromosomal genetic
Yes ma'am.
material. Huh? Mm-hmm.
Yes, that's cope with the resistance and it is in grow independently. Main points, it's independently, and in bacteria cells it is present in many hundreds in one bacteria. Yes, ma'am.
Yes. Так, Kavar, can you tell about
Yes, ma'am.
volutin granules?
Ma'am, what? Volutin granules?
Yes, volutin granules.
Volatile granules.
Volantine.
Huh.
Ma'am, yes, of course I can tell about Volantine granules. Volatile granules, ma'am. Ma'am, volatile granules are interceptional storage form of complex organic polyphosphate, the production of which is used as one of the identifying criteria when attempting to isolate Corynebacterium diphtheria or Lophium medium. Volatile granules are characteristic present in diphtheria and bacilli. And volatile granules, sometimes termed metachromatic granules because of their color reaction with the dyes used in light microscopy, contains polymerized inorganic phosphates and energy rich compound that act as a reverse store of energy and a phosphate. And polyphosphate granules would display the metachromatic effect, appearing red when stained with the methylene blue. And they can also be found in the cytoplasm, saccharochimes, a genus of ascomycete fungi. And
Yes.
yes, ma'am. And volatile granules are intracellular storage of complex inorganic polyphosphates. Ma'am, they are intercellular storage of complex organic phosphate.
Present. In which present?
Yes.
And
Corynebacterium.
Okay.
and
Так, now I think we have WhatsApp group, right?
yes,
Yes,
ma'am.
ma'am.
No, I don't. Have it,
Yeah.
Alina.
We have.
We have.
Yes, we have.
Okay, ma'am.
होगा इतना तो ग्रुप ही तो बस बनाने आए हुए हैं.
वो तो है. Yes, ma'am. We have WhatsApp group.
Yes, to WhatsApp group. Now I will send one picture.
Okay.
Can you see?
Yes, ma'am. We
I
have
send
seen.
it.
Yes, ma'am.
Yes, ma'am.
Okay,
It's
this is
Cytoplasmic granules of
Valentine
bacteria.
granules.
yes.
Yes.
Now you can write Valentine granules and draw. See why it is metachromatic granules? Because the body will take another color and the granules will take other colors. Yes,
Yes,
Yes.
ma'am.
because these granules are mm, storage of nutrients, right?
Yes.
Now you can draw and then you will show me.
Okay, ma'am.
रीना ड्रा करके भेजो यार.
हाँ करने लगे. Yes, ma'am.
मुझे भी भेज दे लीना. मैं भी कर लूंगा.
Okay,
ये
you
ना
can
दे.
show me.
Okay. Wait a minute. Here it is, ma'am. You can see.
Yes. In the both side. Yes. Good.
Okay, ma'am.
Cover here.
Ma'am, just 1 minute. I am making.
Okay. Yes. Good. Very good. Okay, that was volatility granules. Now let's
Yes.
talk about flagella.
Yes. I talk, ma'am. I will talk.
Yes.
Ma'am, actually, flagella are thread-like, hair-like structures that involve in locomotion of cell, and it's also the word flagella comes from flagellum, which means whip, and some special flagella are used in organisms as sensory organs that can sense
Mm-hmm.
changes in pH and temperature. And
Mm-hmm.
it's we talk about specifically functions, then it is a primarily mortality organelle that enables movements and chemotaxis, and they can be either polar or peritococcus, and
Mm-hmm.
on the other hand, and there are other types of flagella, monotrichous, amphitrichous, lophotrichous, and peritrochus. If we
Mm-hmm.
talk about all of them separately, then monocritus is a single polar flagellum, like for example, Vibrio collida has single flagellum, and
Yes.
on the other hand, if we talk about amphitrichous, single flagellum on both sides. For example,
Mm-hmm.
Alcalaegenes facialis,
Mm-hmm.
and Lophotrichus, tufts of flagellum. Tufts mean bunch of flagella on both sides. And
Yes.
for example, Spidellum have a tuft of flagella on both sides. And on the other hand, if we talk about say Peritrochus means a numerous flagella all over the bacterial body. मतलब it spreads all over the bacterial body, not in the
Mm-hmm.
tuft form, and all over the body. So these are the basically types of flagella. If we talk about the structure of flagella, then it has basic three parts: filament, hook, and main basal body. And the filament is rigid and uniform structure that extends from the cell surface, and it contains protein flagellin, that is a base and main component, and composed of protein flagellum arranged in a helical chain, so as to form a hollow core. And if we talk about the flagella found in human body, and which structure will use flagella to move you, and the human cells that have flagella are gametes. For example, sperm cells, it's a sperm also has. So
Mm-hmm.
yes, ma'am.
Yes. Okay, now can you draw these four types of
Flagella?
flagellated bacteria? Yes,
Yes, ma'am, we can.
amphitrichus. Uh-huh. How are
Okay,
you
give.
also? Mm-hmm.
Yes, okay, ma'am. Give us just 5 to 10 minutes. Okay, 5 minutes.
Yes, yes,
Good.
yes.
Yes. Okay. Ma'am,
Yes. Mm-hmm. I'm
no, no.
waiting.
Yes,
Yes,
ma'am.
yes, you
We
can
draw.
draw.
We are drawing. Yes.
Uh,
Hello, ma'am.
yes.
Ma'am, I have done.
Mm-hmm.
Ma'am, it's the first one is at trichus. It's have doesn't
Yes.
have any flagella around, and this one
Uh-huh.
is monotrichus. It has one
Mm-hmm.
flagella, singular,
Yes.
and it is amphitrichus. It has a single flagella on both sides,
Mm-hmm.
and it has a cephalotrichus. It has a bunch of bacteria, bunch of sorry flagella on both sides. It's called tuft.
Mm-hmm.
Yes,
Yes.
and then peritrichus. It has flagella all over around,
Mm-hmm.
and
Yes.
then we call it lophotrichus. It has tuft of flagella on its one side.
Mm-hmm.
Yes, ma'am. This is
Okay,
types of
good.
flagella. Okay, ma'am.
Havar, have you finished?
I finished.
Mm-hmm. Yes, good.
Ma'am,
Arthritis, mono.
Atricus, monotrichus means only one flagella. Mono means one.
Mm-hmm.
Amphitrichus, they have on two flagella on both sides. And Cephalotrichus, they have many flagella on the both sides, with eight flagella. And Peritrachus, they have flagella surrounded all over the body, and Lophotrichus, they have one side flagella. They have many flagella on one side.
Okay. Do you know the detection of mortality? Detection of mortality.
Detection of
Bacteria.
mortality, ma'am.
Yes, yes.
Detection of mortality.
How we can?
In fresh culture, if we take a fresh culture of bacteria,
Yes.
then we usually microscopy most accurate way to determine bacteria mortality and a hanging drop method.
Ma'am,
It is commonly used bacteria technique. For example,
Yes.
motile bacteria swarm and give a diffuse spreading growth that is easily recognized by naked eye. And usually there are many different methods for detection of bacterial mortality. For example, method is touch a straight needle to colony of young culture growing on agar medium, and then stab once on deep, uh, inch in the middle of tube, 1/3 or yeah 1-2 inch in the middle of tube. Then we incubate at 35 to 37 and examine it up to for 7 days. We keep this on this temperature 35 to 37. Then we observe for different zone of growth, like flirting out the line of inoculation. That is usually easily we can see on our naked eye. It's a medium culture of detection, and mostly two mortality tests we have done: small bowel endoduodenal manometry, and these tests provide information regarding muscle activity of the stomach and small bowel during fasting, for example meal, and during sleep. Yes, ma'am.
Good. Yes, that is the detection of mortality. That means we can
Yes.
detect them by microscope.
Patriarchus.
By microscope,
Yes,
and
ma'am.
also we can cultivate them on
In agar
culture.
solution
Yes.
medium. Yes.
Then we can see how they
Swarm
will
of
grow.
bacteria.
Yes, good.
Yes.
Cover. Now your turn.
ma'am, what should I tell?
About capsule.
Capsule, ma'am.
Capsule.
Capsule bacteria.
Yes, ma'am. Ma'am, the bacterial capsule is usually a hydrated polysaccharide structure which covers the outer layer of cell wall. And in most bacteria, it is composed of monosaccharides linked together by a glycoside bond. However, amino acids and protein carbohydrate capsules have also been described. Ma'am, now it's what capsule is made up of bacteria. The capsule is composed of polysaccharides that cover the cell wall, which is made of peptidoglycan and teichoic acids, characterizing the classic gram-positive structure. And it acts as a principal antiphagocytic and protective elements that prevents the access of the leukocytes to the underlying cell wall elements. Now. And the role of capsule, the capsule helps the bacteria adhere to the surface, camouflages the bacteria from the immune system by mimicking the host tissue, and make the bacteria resistance to complement invasions. A capsule protects cell from dissociations and toxic metabolites in the environment. For example, heavy metals and free radicals. And capsules and the capsule is found mostly common among gram-negative bacteria, Escherichia coli, and in some strains, Neisseria meningitis and Klebsiella pneumonia. In these bacteria, capsules are mostly found.
Yes.
And the bacteria which in the respiratory, ma'am, in the respiratory tract and oral cavity, there are several species without capsular material on the surface. Means some bacteria are present which do not have capsules. For example, gram-negative Haemophilus influenza This in this bacteria, in this gram-negative bacteria, capsules are not present. Ma'am, the two main function of the capsule in bacteria, it protects the cell from being engulfed by white blood cells.
Yes.
Yes, and and all bacteria secrete some sort of glycocalyx, an outer viscous covering of fiber extending from the bacterium, and extensive tightly bound glycocalyx adhering to the cell wall, which is called capsule.
Yes.
And in the biology, a capsule refers to membranous shed that surrounds a bodily organ such as kidney or fibrous tissue that surrounds a joint. This is the biology of capsules, that how it surrounds the bacteria. In microbiology, the capsule helps protect bacteria from phagocytosis as well as from desiccations. It also helps to adhere the surface of the cells. Yes, ma'am. And the function of capsules in prokaryotic cells, ma'am. I'm now telling the function of capsules in prokaryotic cells. Ma'am, first of all, the capsules helps prokaryote cling to each other and to various surfaces in their environment, and also helps prevent the cell from drying out in case of disease causing prokaryotic that have colorized disease, causing prokaryotes that have colonized the body of a host organism. The capsule or small layer may also protect against the host immune system. In the bacteria which have capsule, it protects against the host immune system, the host which have immune system in prokaryotes. Yes, ma'am. And
Okay, good. Now, how we can detect capsule?
how we can detect capsule, ma'am?
Detection of capsule.
Ma'am, the
Ma'am,
Do
I
you
can
know?
tell.
Yes.
Yes, ma'am. Yes.
no,
Ma'am,
you can.
we usually use India ink, that's gram crystal violet, and bacterial capsules are non-ionic, and so it cannot be either acidic or basic stain. So we use other methods to adhere their surface. Therefore, the best method to stain background using an acid stain, and to stain the cell itself using a basic stain. So we use India ink and gram crystal violet.
Yes.
Yes, and the main why we use capsule stain show, and the main purpose of capsule stain is to distinguish capsular material from bacterial cell. So capsule is a gelatinous outer layer secreted by bacterial cell, and surrounding adheres to the cell. So most of the capsule are composed of polysaccharides, but some are also polypeptides composed of poly. And
Yes.
this is a negative staining technique that is essentially used to identify presence of capsule. So because of its acidic nature, India ink, we also India ink or yeah, Congo red nigrosine stains. Yes, it's we usually use for the visualization, and capsule is a slime were visualized electromicroscopically in Rhodos Pseudomonas capsule strain. Yes, ma'am. It's we
Yes.
usually use. Yeah.
Yes, you are right. That here we can use them Indian ink
India
or
ink or
negative.
Congo red, nigrosine, stain the background dark.
Yes. Now I can send the capsule
Okay.
staining, which you also have to draw.
Yes. Okay, ma'am. Okay. Okay, ma'am.
Ma'am.
Yes, ma'am. I
Okay.
have also done. Ma'am, here it is. Can you see?
Uh-huh. Yes.
It's a background which I draw black
Mhm.
background, dark, and then it's a capsule, and here it's
Yes.
a bacterium which is central, and
Very
here
good.
is filling process. Okay.
Yours is better. However, can you compare your diagrams?
I am actually very bad at drawings. It means very, very, very bad at drawings.
No, you can draw.
Yes, but ma'am, I don't have too many pencils now. I have only red pencil. This one.
मुंह से ले जाएंगे.
तू अली ना मार खिलवाएगी हमें. इंसान की बच्ची बन के
Okay,
बात
okay.
कर.
Yes, ma'am.
But Alina, yours is good.
Ma'am,
Really
she
good.
is our topper bachchi.
She is what?
Our topper bachchi means intelligence to hard work.
You too.
Yes, ma'am. He is also very intelligent.
Ma'am, your picture is very beautiful. Where did you capture that?
Hello, ma'am.
You, yes, yes, you too. That's why you are attending the class while rest of.
No, ma'am. He asked, your profile picture is very good. Where you captured from?
Yes, ma'am.
My that photo.
Yes, ma'am.
Yes, ma'am.
I went to India. That day I took this.
Okay, ma'am. Red
Ma'am, which
Mosque.
place is this?
It's what was the name?
Ma'am, come to Pakistan, please. We will show you around the Pakistan.
Yes, I hope in the future I will go.
Yes, ma'am. If you and to come, I
Yes,
will
ma'am.
just call
Pakistan,
me, ma'am.
northern
I
areas,
will pick you
Kashmiri
up.
sides, Patriata. हाँ तो मैम को
Mhm.
घुमाना.
You will love the Pakistan, ma'am. Ma'am, the dupatta is very beautiful you are wearing.
Okay, I will
लड़कियाँ हमारे.
think about,
मक्खन बचा है थोड़ा सा.
and everything it will be fine without coronavirus.
Yes, ma'am.
That
Ma'am,
is the problem.
you know, no, for doctors, online studies like nothing.
I know.
Yes, ma'am.
I can understand.
We are just seeing something and we are just studying by our own.
Uh-huh.
Yes, ma'am. That's why
And
most of the students don't join. Don't join. Don't join. Sorry.
I don't know why they don't want.
Ma'am, because they, ma'am,
Yes, ma'am.
basically when we started, it's our third semester. And we
Mhm.
just took 3 to
Mm,
4 classes offline.
Mm,
After that, we are taking online classes. And that's why all are depressed.
okay, I understand. But what we can do? Whole world studying like this only.
yes, ma'am. When Corona will go and we normalize our life.
Yes, yes, yes. I hope so.
But malls are open.
Yes, ma'am.
Yes, I also don't know. I cannot understand this.
Why they cannot
Ma'am,
open the
every
university
country
while malls are
closes
open, ma'am?
educational institutions.
Garages are
Pakistan,
open.
Pakistan also closes its educational institutions, but malls, marriage halls are on the other side open. Everyone targets its education.
Uh-huh.
हमने
Yes.
literature. Yes, ma'am. Malls, marriage halls, and other restaurants also opens here, also Pakistan, but not education institutions.
Uh-huh.
Yes,
That is the
ma'am.
problem.
Yes,
It,
everyone
it
targets
seems, yes,
studies.
it seems they wanted to broke education system.
Yes, ma'am.
But we will try to do our best.
Yes, ma'am.
Yes, hope so.
Okay, I can send one more picture, and then we can
Okay.
discuss
Free.
about that. And then you may be
Free
free.
to go.
Uh-huh.
अगली क्लास
Thank you,
कब
ma'am.
12:40
Okay, this is the last picture. You can draw this, and
Okay,
then we
ma'am.
will.
End lecture.
The structure of bacteria.
Done, ma'am.
No, ma'am, just 2 minutes.
Mhm. Mhm. Okay, I can see.
Thank you, ma'am.
Yes, ma'am, I have.
Mhm.
Ma'am, it's bacterial cell, which is its pilus, its flagellum, its DNA, and these are cytoplasm.
Yes. Okay, we forgot about pili, yeah, to talk about this structure.
Pilus?
Yes.
Yes, ma'am.
When you say, I remember.
Yes.
Okay,
Ma'am,
this is.
it's a hair-like appendage, as you can see, found on the surface of many bacteria and archaea. Term pilus and fibria fringe can be used interchangeably through. If we talk about its function, it's basically our protein structure, and that extends from as a bacterial cell and envelop
Uh-huh.
for a distance up to 2 micrometer, and
Yes.
they function to attach cell to surface.
Mm-hmm.
Yes, and
Yes.
it's a hair-like structure on the surface of bacterial cells, resembling like a hair.
Uh-huh.
And fibria and pili, both are thin protein tubes and originates from cytoplasmic membrane of many
Mm-hmm.
bacteria, and both are able to stick bacteria to surface, but pili are typically longer and few in number than fibria, because it's a number, it's usually 2 micrometer. So it's found in virtually all gram negative bacteria, but not in many gram positive.
Mm-hmm.
Yes, ma'am. It's a main function.
Oh,
And
yes. The main, as you say.
these hairs are also involved in bacterial conjugation.
Mm-hmm. Mm-hmm.
Yes,
Yes.
and it's
There
allow
can be sex
also the
pili and
transfer of DNA.
adherence
Uh,
pili.
yes, and it's also allow transfer of DNA between bacteria
Mm-hmm.
in the
Yes.
process
Yes.
of bacterial conjugation.
Mm-hmm. Yes. Good.
Yes, ma'am.
And all
And
this
it's we can
structure are.
yes, and we can also say sometimes sex pili, because it's allow the exchange of genes via the formation of mating pairs.
Uh-huh. And all
Yes,
these structure are divided into essential and non-essential.
yes,
For
ma'am.
essential, that structure, without this, they cannot live.
Yes,
It is cell wall, cell membrane, cytoplasm, and nucleus.
yes, ma'am.
Nucleoid. And other
Nucleus.
like flagella, capsule, polyethylene granules, plasmids, they can live without these structures.
Yes, ma'am. You are right.
And one more thing about cell wall. Sometimes bacteria can lose their cell wall in the presence of antibiotic or enzymes. And at that time, we are saying that it is L form of bacteria.
Yes.
Okay, L form. Uh-huh.
Okay, ma'am.
When they will lose their cell wall, but they can multiply. And when we will stop giving antibiotics, they again will synthesize their cell wall and can live. Okay. Now,
Yes, ma'am.
now we can. Now you are free because we discussed all about
Yes, ma'am.
cell
Yeah, ma'am,
structure.
also give us next topic so we can prepare.
Okay, next will be about sterilization, the types of sterilization,
With
disinfection.
bacteria.
What?
Thank you, ma'am. Thank
Sterilization
you.
process according to bacteria.
Yes, yes, yes.
Okay.
And in general also.
Okay, ma'am.
And then nutrition,
Okay.
nutrition and culture media. Okay.
Okay, ma'am.
Types of sterilization, nutrition,
Nutrition
classification of
and culture
bacteria
media.
by nutrition and culture media, requirements for culture media, and that's all.
Okay, ma'am.
Okay, bye.
Okay.
See you
Bye,
next
ma'am.
class. Also there will be control work,
Yes.
but till the next class I will inform you how it will be. Okay.
Okay, ma'am.
Bye.
Okay, bye.
Там где-то что-то.